Welcome back, students. This is a second video lecture on chapter 1. Today we're going to talk about cell classification and the history of cells. The agenda for today's video lecture is the following. If I can get my cursor working. Here we go. We're going to look at cell classification first. You'll be surprised that there are a few different ways to classify cells. Then we go on to look at the similarities and differences between prokaryotes and eukaryotes. Thirdly, we're going to look at the theory of endosymbiosis, a very proven mechanism that helps us understand the history of present-day cells. Then we go on to look at cell anatomy and terminology. Very important in a class like this, where using the right terminology at the right time conveys the right message to the listener. Then we look at model organisms, sometimes confusing for students, so I'll try to make it as easy as possible. And then lastly, this goes with a figure that's present in most cell biology books, which relates the size of the DNA content of a cell to the species to which that cell belongs. Is there a relationship? Do humans have the most DNA of all creatures on the planet? Well, that's called the C-value paradox in molecular biology. So let's get started with number 1. So I think my cursor is ready to run, and let's look at cell classification. So the most fundamental type of classification that you should be aware of throughout this book is that cells fall into 2 buckets. So if you discover a new cell underneath a rock, all you have to do is look under the microscope to see if inside that cell there is a nucleus. If there is a nucleus, then it belongs to the eukaryotic branch of the tree of life, right? And we call these classes. So it belongs to the eukaryotic class. If the cell is missing a nucleus, then it's a prokaryote. So you just have 2 choices in this case, no more than that. Let me pull down a pen because I need to use that. So prokaryotes and eukaryotes are just the 2 classes of cells that exist on the planet. There's nothing else that we know of, right? So either you're left or you're right. You can't be halfway. The next classification system that we want you to be aware of are domains. The way I explain this to students is internet has a domain. Well, guess what? Life has had domains for much longer. So how do we classify cells into domains? These are based on mostly biochemical differences. So these differences aren't obvious from the outside, but when you start examining using modern technology, some of the biochemistry of cells, you discover that they fall into 3 discrete buckets, right? You have the archaea, the ones in the middle. And these are the cells that are prokaryotic. So let's draw a line down here. Let me get my cursor back, it likes to disappear. So these belong to the branch of prokaryotes, but their biochemical nature is completely different to the second branch of prokaryotes, the bacteria. So prokaryotes are divided up into 2 discrete domains. Now, what's the difference between bacteria and archaea? Well, in your gut, E. coli, the one that gives you food poisoning every now and again, that's an example of bacteria. So most bacteria we know of as disease-causing organisms. The second branch, the second domain, archaea, is those creatures that live in harsh environments. So in my mind, Yosemite or Yellowstone or any other harsh place like the top of Mount Everest comes to mind. So if you go to Yellowstone National Park and you find those hot water soups where the geyser is going off and it's really hot to touch, or they're acidic or they're very salty, well, inside those ponds, you'll find archaea. So they like to live in harsh places and extreme environments, right? So that's where you normally find them, although they're around you right now too. Maybe some of them are on your skin. And of course, the third domain of life is the eukaryotes, right? Or eukarya. So that includes all eukaryotes. So eukaryote is easy because this includes all cells that have a nucleus. So you see the relationship between the class and the domain. Let's move on to the last one. And the last one is controversial because different scientists like to use different numbers. Some think it's 5, some think it's 6. In some places you'll see 7 kingdoms, right? But let's stick with 5 and 6. So when it comes to 5, what they do, the scientists, they divide the eukarya into 4. So we have the first branch are the protista. This would include the amoebas of the world, right? The planaria of the world. And mostly single-celled creatures, not exclusively, but mostly. They're microscopic, hard to see. Although the other end of the protista spectrum would be the giant kelp that we find off the coast of Monterey, growing to 100 yards. So those would be protista too, right? And protists can cause a disease, like Plasmodium is the disease that causes malaria, or the parasite that causes malaria that's carried by the saliva of mosquitoes, and sleeping sickness and yellow fever and things like that, okay? The next one are fungi. So the fungi, the kingdom fungi belong to eukarya and eukaryotes. And this would include athlete's foot, toadstools, mushrooms that you eat, right? So fungi are those types, that kingdom. Then we have the kingdom of plants. So those are also eukarya, and these are photosynthetic creatures that can manufacture their own food. So all plants belong to the kingdom Plantae. And then finally, we have the animal kingdom, to which we belong. And these will be all creatures that are multicellular and that can move around. So we'll learn more about that in the future. So 4 kingdoms belong to eukarya. The other 2, in this case, or the other 1, according to some textbooks, it would be the kingdom bacteria, right? But I like to go with the double meaning, so we go for the 6th kingdom definition. So bacteria are bacteria, and archaea are archaea. And that will give us our 6 kingdoms of cells. Okay, so it's easy to forget, and the one that students normally have a hard time with is number 3. They forget that, right? But once they remember number 3, then they always get stuck on number 4, fungi. It's the obscure one. But try to remember the 2 worst ones, 3 and 4, and that will bring back the entire list, okay? So it's very important that you keep in your mind, whenever answering any questions in this class or into the future, where cells are belonging to that particular classification style. Okay. Let's move on to the next question. The next concept, sorry. In this one, we're going to compare prokaryotes and eukaryotes. If you remember the video that I made for lecture 1, we talked about the definition of life and the cell theory. So they apply to this whole slide. So what you see here are living cells, right? So they all have to satisfy Mr. Gray plus, and they all have to satisfy the cell theory. So that's what they have in common. Other things they have in common, yes, there are certain biochemical reactions and certain types of biochemical pathways that are common to all living things. And we discussed that in the unity of life. What we're trying to do here is look deeper inside these cells and see if we can find any differences. And I've listed just a few to get you started, right? So the obvious one is that prokaryotic cells, they don't have a nucleus, whereas eukaryotic cells do have a nucleus. We mentioned that earlier in a previous slide. The second most common definition of the difference between the 2 in most books is that prokaryotic cells, when you look inside them, they don't have any membrane enclosed organelles. That means that they don't have internal structures that are wrapped themselves in a wrapper of membrane, right? Whereas eukaryotic cells have a lot of these. So let's put an X here again, if I can get my cursor to work for me. Where are your cursor? Oh, it's up here now. There's the cursor. So there's an X there and there's a check mark here, right? So an example of membrane enclosed organelles will be lysosomes, Golgi apparatus, endoplasmic reticulum, smooth and rough, mitochondria, chloroplasts, peroxisomes. Did I mention them already? And then there's many more. There's vacuoles and this and that and the other, right? Those are restricted more or less to the eukaryotic cells. Prokaryotic cells seldom have these structures, although we have examples of some that do. By far, they don't. So we use that very simple criteria there. The next one is sort of relative. In general, prokaryotic cells are small, whereas eukaryotic cells, in comparison, are larger, although there's some overlap that you can see in the numbers, right? About 10 times smaller on average. So these are about 50 micrometers, and these will be about half to 1 micrometer, right? So you can see there's a 10, in terms of cell size, there's a difference in the length, generally, right? So that's important to realize. The DNA within prokaryotic cells is normally circular and is just parked in one corner of the cell called the nucleoid, right? In contrast, in eukaryotic cells, the DNA, most of it is in the nucleus and it's linear. But we mustn't forget that eukaryotic cells, all of them contain mitochondria, right? And mitochondria contain their own small pieces of DNA. And that DNA in the mitochondria looks like this DNA in its form and function, right? So that sort of gives us a clue as to what's coming up in the next slide, where we talk about where did mitochondria come from. And of course, the same thing with chloroplasts. So plants and some other protists have chloroplasts. And those chloroplasts also have a small DNA content of circular DNA. So they also resemble the DNA that you find over here. And there's a bit more to the story than that. But for this particular slide, that's what you need to know, right? So can you make a list of 4 or 5 differences between prokaryotes and eukaryotes? Thank you. Let's move on to the next slide. This slide talks about the theory of endosymbiosis, a very popular and much supported theory in biology. So it has lots of evidence, right? And I'll show you the evidence when I click on the next half of the slide. What does this theory say? It says in the long history of life on this planet, which has so far, we believe, lasted about 3.5 billion years, not million, but billion, things have happened where evolution has taken place and a single species of original cell, whatever that was, split up into 2, and then that split up further and further and further, and it became just like a giant tree, with some branches going extinct and others going forth. We are one of those branches, humans, the human species. So our chimpanzees, they're another twig on the same branch as us, right? So each endpoint is a species at the moment. But let's go back in the history. Some time ago, about 700 million years, maybe 800 million years, 1 billion years ago, we're not quite sure, but some time ago, small cells were eating large cells for food. So a small cell would eat a large cell. As the cells got larger, they had a problem with not enough membrane to allow them to exchange chemistry with their environment. So let me put this in a different way. If you think of an elephant, why aren't elephants any larger than they are today? Without going into too much detail, right? It has to do with the amount of skin they have compared to the environment in which they live, right? So an elephant, if it got larger, if it got any larger than it is now, would have too much body and not enough skin, because the skin doesn't keep up as the body begins to grow in size. So the same thing you can apply to this cell here, the one in blue, the first one. It has enough skin, membrane, to supply enough nutrients and remove enough waste from that cell to keep the inside working perfectly. But as the size of the cell grows, then the proportion of skin to the cytoplasm, the inside, begins to become unfavorable. And the skin problem, or the surface area to volume problem, as we call it, will become a physical and chemical problem, and life has to then overcome that. And what do we do? Well, one thing to do is to increase the ruffles on the external membrane. By having little fingers protruding from the skin, the amount of skin now covering my hand is increased. Therefore, it allows the volume of my hand to increase further. So that's one solution, right? The second solution is to divide the cell into compartments. And each compartment can then do its own biochemical reactions without interfering with the different compartment. It's like us in a library, right? If everybody in the library is in a single room and we're talking to each other, it's very hard to hear any particular conversation. So to overcome that, what we can do is construct a library with lots of rooms. And if you and I want to have a conversation, we just go into one room and we can have our conversation by shutting the door, and we won't be disturbed by the other voices in the other rooms of the library. So this is what the nucleus is. Think about it. The nucleus that eukaryotic cells have may be a result of their larger size. Because I told you on the previous slide that eukaryotic cells are 10 times longer than prokaryotic cells. Being 10 times longer and a cube shape makes you 1,000 times bigger than an equivalent cell that's 10 times smaller, right? That's mathematics. And I think your TA would go through an exercise to show you that very shortly in discussion. But let's stick to this point. As the cell becomes larger, it makes sense to start putting internal compartments in. So you can see here, the nucleus of this cell formed when it became a certain size. And that nucleus then trapped the DNA and the biochemical reactions that are centered around DNA. Therefore, leaving the cytoplasm, the area outside that, this area here, to then perform other reactions independently of the reactions inside the nucleus. So larger cells tended to require the evolution of a nucleus. So once the nucleus evolved, then we had the existence of eukaryotic cells for the first time on the planet. Now, we're not sure which came first, the nucleus or the organelles, but at some point in the history of life, these larger cells, this one here in blue, started eating the smaller cells, this one here in green, which I'm coloring in now, right? So there you go. And the green cells were food for the larger cells. And the larger cells will completely digest the green cell by taking it inside their cytoplasm and then breaking it down and using its molecules for food and energy. Now imagine that process being unsuccessful, i.e. you eat the smaller cell, the larger cell eats the smaller cell, but it doesn't digest it. And it sits around inside the cytoplasm. Now, you could say that could be harmful. Yes, in most cases, that would lead to the death of both cells. But if the mutations happen in a favorable way, which they can, if you have enough of them, there's a chance that the green cell that was ingested may continue to live happily inside the cytoplasm of the blue cell. And that relationship could be then passed on to future generations of blue cells. So if the green is multiplying inside the blue cytoplasm, and then the blue splits in two, and there's also always going to be some green in this daughter cell and some green in this daughter cell, and then that process will continue again and again. And that's what we believe happened. So that is called the theory of endosymbiosis. The green cell over here in this figure was endocytosed, taken in to the blue cell, but it didn't die. It lived happily inside. So the two lived in harmony. That's your symbiosis, endosymbiosis. That's your endosymbiosis theory, right? we believe that all eukaryotic cells came from this original sequence of events. So any eukaryotic cells that didn't have an internal nucleus and swallow the green cell, we can't find them, right? So they must have been outcompeted by natural selection. So all the eukaryotic cells in the world acquired this internal passenger that evolved slowly to become mitochondria. Right, so your mitochondria, my mitochondria, and all other mitochondria in all eukaryotes is actually an ancient bacteria that took residence inside our cytoplasm. All right, and that gave rise to what we see here, modern day animal cells. Modern day animal cells. And also, you know, fungi also have mitochondria, so do protists, so do plants. But something strange happened to one of the branches, this one here. So this branch, it had the mitochondria, let's call them M right there, right? These are mitochondria. But this sequence of, or this lineage of cells, it acquired later on a photosynthetic bacteria. So they did endosymbiosis again, and they picked up a photosynthetic bacteria. So these were bacteria that could absorb sunlight and use that to make their own food. So these cells accidentally picked up these, or on purpose, and they started living inside their cytoplasm too. So now this branch of the eukaryotes had two passengers, the mitochondria bacteria type, and then the photosynthetic bacteria type. And over time, they became the norm, and today we call them plant cells. So plant cells have inside them not only mitochondria, but they have the photosynthetic bacteria, which we call chloroplasts. So that in itself is amazing. How do we know this? Well, let me pick up my cursor over here and show you how that works. What do I need to press? I need to press the home button and get back to my cursor. All right, let's clear all that. So on the next half of the slide down here, let me remove this figure, this graphic. So I have something hidden underneath for you, right? So the evidence that we have to support this theory, because nothing in science is valid until you can show some kind of a support for it. Mitochondria and chloroplasts, these internalized bacteria, they should have some chemical signals of their own ancestry. And indeed they do. So if I look at your mitochondria, or if you were a plant and I looked at your chloroplasts, biochemically, I would find that they have telltale signs that they used to be ancient bacteria. The first thing is they both are surrounded by a double membrane. How does that work? Well, think about it. I'm the large cell, I have a single skin. And let me use something here. This is a bacterial, a smaller cell from the past, right? Then if I swallow this, and as it's got its own skin, the blue skin, and my hand has the brown skin, right? As I swallow this and I wrap around it like that, then it becomes inside me. You can see you have the blue skin on the inside that belonged to the bacteria, and then you have a wrapping of brown skin that was picked up as the cell came into the cell like this, of brown skin, right? So now you have two layers of skin. So we find that in both mitochondria and chloroplasts. Indeed, the inner skin has the telltale signs of the chemistry of bacteria. The outer skin surrounding the mitochondria and the chloroplast has the telltale characteristics, chemical characteristics of the skin of our cells. So this is one line of proof that this process did happen in the past. The second thing is the way that the mitochondria reproduce inside our cells. They don't follow the normal way that other organelles and other structures reproduce, right? They're made in one location and delivered to their destination. And what mitochondria and chloroplasts do is they reproduce in a similar fashion to bacteria that live today. So what would happen is that if you need more of these mitochondria, say this is a mitochondria and I'm the cell, if you need more of these, this will just get larger and larger and split in two. Now you have two mitochondria. If you need more, each one will get larger and larger and split. So that's how they reproduce. Very similar to the way that bacteria reproduce. They don't reproduce like our cells. So that's another line of evidence, right? When you look inside the mitochondria or chloroplasts, you have DNA. And that DNA is circular, just like we mentioned earlier in prokaryotes. Not only that, the signature of the DNA is very similar to modern day bacteria than it is to our DNA. So there's more proof that maybe these structures were ancient bacteria. And then lastly, inside the mitochondria and the chloroplasts, they have their own ribosomes. What are ribosomes? Well, they're just machines for making protein. So we have in our cytoplasm millions of ribosomes, but guess what? There are ribosomes that are also trapped inside the mitochondria. And when you look at their chemical signature, they resemble, those ribosomes, they resemble modern day bacteria, whereas our ribosomes are different. They're larger. They're made of different combinations of proteins. So there's more evidence to suggest that these were really ancient bacteria that started living inside our cells. There you go, the theory of endosymbiosis in about 10 minutes. If you want to watch the video, that's a great way to have a animated perspective on the same content. All right, we're more than halfway through. So let's carry on to the next slide. This is where we're going to learn about terminology. So when you communicate with each other, you're using the right terminology. If I said I'm catching the train and I get on the bus, that's not good, is it? So I better tell you I'm getting on the bus and not the train. So the same thing here. So take a cell. So you can take a bacterial cell. Let's do the bacterial cell first. Let's call it what it is, a prokaryotic cell, right? So that top cell in brown has no nucleus. So it's a prokaryote by definition. So let's look at that. So here's the cell itself, right? And how can we then break it down into compartments? Well, we know that it's surrounded by a membrane. The membrane is right here in this light brown color. Then it has one or more cell walls on the outside, like an overcoat. You can see that in purple and dark brown. So these are overcoats. But inside the brown envelope, the internal membrane is the gel-like material. And we call that, all of that, the protoplasm. Protoplasm, right? Proto means living or lifelike. Plasm means substance. So bacteria, the cells are composed of a skin, the membrane with outer layers of cell wall. Then inside the skin is the gel-like material, which we call protoplasm. And that protoplasm can also be referred to as cytoplasm. And that's about it. That's what bacteria have, that arrangement of there, right? So get used to that. So cytoplasm of a bacterial cell, to me, means everything inside the membrane, the plasma membrane of that cell, everything, including the nucleoid where the DNA is, the ribosomes, and all the other structures, okay? So that's very important. But what we want you to learn are the other terminologies on this slide too, and they pertain to a eukaryotic cell. So the bottom cell is a typical eukaryotic cell. To me, by looking at it, it looks like an animal cell. I can tell by the structures inside it, and you should be able to do that too in the future, I should say. So let's go back here and let's erase this. And let's get rid of this. And now we're going to apply the same analysis to a eukaryotic cell, okay? So let's put this away. So eukaryotic cell, as you can see down the bottom, the blue thing, is a cell. And in the case of animal cells, they have a membrane, but we don't have a cell wall. Our cells are squishy. They don't have an overcoat, right? Whereas fungi and plants, they will definitely have an outer coat, a cell wall. But we're talking about animal cells. So an animal cell has a membrane, a plasma membrane, and then inside that membrane, just like with the bacteria, it has protoplasm, all the living matter, right? That protoplasm can be divided into the area inside the nucleus. So the nucleus will be number 2, and then everything outside that. So everything outside the nucleus is called cytoplasm. Between the plasma membrane right? And the nucleus, everything in between is called cytoplasm. Now, inside the nucleus, there's a structure, number one, and that is an area that stains densely under certain conditions. And we call that the nucleolus, the nucleolus. What is that doing? Well, one of the worksheets that you'll be getting will help you relay that information. But the nucleolus is a hotspot for certain types of biochemical activity where you're making ribosomes, right? But don't worry about that. It's a hotspot. So the nucleoplasm is the nucleus, and inside that you will find one or more nucleoli. But the point I'm trying to make is with the cytoplasm. So the cytoplasm can itself be divided into two regions. The first regions are the organelles. And that would be everything that you see there, number 12, number 10, number 5, number 9, number 4, number 8, number 6, right? So all those structures would be called organelles. So would be the ribosomes that are also present there in tiny sizes, millions of them. So all those are called organelles, and they're suspended inside the cytoplasm, right? But what about the area that doesn't include the organelles? The light blue area inside that cell? Well, that's called cytosol, right? So that's like the gel-like matrix that surrounds all the organelles between the nucleus and the cell membrane. So the point of this slide is please use cytoplasm when you mean everything between the nucleus and the plasma membrane. But use cytosol when you're talking about things that don't include the organelles, right? So if I was talking about something here, then I would say cytosol. If I was talking about something here that cuts across both of them, I would say cytoplasm. So this is very important. And the same thing will apply as we get into deeper and deeper chapters. Some of the words are very similar, right? Cytosol, cytoplasm, centriole, centromere. Oh my God. There's hundreds of words that start with the word CEN in cell biology. So you have to be very competent at using the right word at the right time. And of course, we will correct you should you be using the wrong terminology so that you can become masters. All right, let's go to the next slide. So the next one is very quick. It's about model organisms. Students have a hard time understanding model organisms for the following reason. It's an abstract idea. But let me give you an analogy. Say you were learning how to fix cars. You wanted to become a car mechanic. And that's your job, right? So you open a garage and people bring their cars in, and you don't specialize in any car. You can take any cars, right? So what do you do when you're learning? Well, you could learn on different cars and share that knowledge with other textbooks and garages, right? But it makes sense if everybody works on one model car at different garages and shares that knowledge. Hey, look, I fixed a radiator on a Ferrari, and you're working on a Lamborghini, and you say, well, I can't find that part on my Lamborghini. Then you get confusion. But if all the garages in the area agree to work on one car to learn how to fix a radiator, then we can share that knowledge. So if everybody starts to work on a Ferrari, I'm working on a Ferrari, you're working on a Ferrari in your garage, then we can compare notes, right? Now, using a Ferrari is pretty expensive to learn on. I'd rather use something cheaper like a Toyota. So expense is very important, right? The size of the car is important because you have a big, big limousine. It's not going to fit in your garage. So other garages may be larger or smaller. So what scientists did in the past, as it says here, this is more appropriate for the past, when technology was pretty limited, in order to share knowledge, they came up with a standard set of creatures that they all agreed to work on so they could share knowledge without having too much complication, right? So if I'm working on a Fiat, or did I say earlier something like a Datsun or something, a Toyota? So if I'm working on a Toyota car and you're working on the same Toyota car, well, it's cheap to buy the Toyota and it's going to be fixed in the same way. So if I'm working on the radiator of the Toyota and you're working on the radiator of the same Toyota, and you discover how to fix it better than me, you can easily share that knowledge with me, and I can see my car and apply it too, right? So model organisms were chosen in the past, about 200 of them, with these being the most favorite. that scientists worked on in different labs around the world so they can share knowledge. So what prokaryote did they choose? They chose E. coli, the bacteria that lives in your gut, to represent all prokaryotes, right? There's millions of prokaryotes out there, but they chose E. coli as the standard, like our Toyota, right? So if you did research on E. coli, you could then share that knowledge with other scientists working on bacteria. So this was chosen as the prototypical model organism for studying. Bacteria are easy to maintain. You can grow them up overnight into billions. You can manipulate them. There's no ethical problems. Nobody complains, right? So that model organism was used a lot in the past. How about a yeast? Well, we chose this yeast over here, Saccharomyces cerevisiae. It's the yeast that we use for brewing beer or making bread, right? So that was chosen by scientists to represent all fungi, right? They didn't choose a toadstool, or they didn't use aspergillus or stuff that grows on your skin, right? They chose the baker's yeast. So that was our prototypical model organism, because everybody had it around the globe. You could buy it from a local baker, because bakers use it all the time, or breweries. So it's just cheap to obtain, right? So you don't have to spend a lot of money. So you can see what's going on here, right? There are certain patterns: cheapness, ease of maintenance, and manipulability, right? So if you can do all these to a model organism, then it becomes a favorite for many labs to do. How about a plant? Well, Thalliana here, Arabidopsis thaliana, is a small 1-inch tall weed that we chose to represent all the plants of the planet. So that's their representative, right? So Thalliana is a plant model organism. Then for animals, because we did a lot of research on animals, we chose lots of animals from different strata of the animal kingdom, right? So we chose a fly, and the fly that we chose was Drosophila melanogaster. Drosophila is the fly that lives on your bananas. You can see it flying around in the supermarket. It's a tiny, 16th of an inch, maybe, fly. And they're easy to maintain. And within 2.5 weeks, you can have a new generation of thousands of flies. And their mutations are easy to monitor. They have red eyes, white eyes, blotchy eyes, long legs, short legs, wings that are folded, wings that are straight, right? Wings that are broken. So we can easily use them for following genetic manipulations. So that was the favorite creature that was an invertebrate, that didn't have an internal skeleton, right? Then we started looking for model organisms in fish, and we came up with the small goldfish-like, it's not a goldfish, I forget the name, what it is. It's like a midgeon, right? A small fish. And we use that to study the development of aquatic animals, animals that live in the ocean, right? So that became our prototypical model organism right there. Because of the size, 1 inch is better than 10 feet across. And they reproduce very quickly. They produce hundreds of babies. So that's easy to maintain in a lab cheaply. And they can be easily manipulated for their development and other stuff, right? We also, and they have bones inside them, just like we do. We also used another invertebrate, the worm, C. elegans, right? So C. elegans is used beautifully because it has exactly, let me get the number right, 959 cells in the adult. And we know exactly where each cell is going to go out of those 959. So it's a very good system to study development. What happens if there's too many cells? What happens if you remove this cell? What happens if that cell doesn't take up its rightful place? So we can then see how our bodies grow by learning from that worm, right? So you can see model organisms help us learn about ourselves, and also maybe to develop treatments for those diseases in that organism, and then see if those same treatments can apply to human disease. So we use the model creatures to understand biology, but also to understand how we work. The mouse over here, the mouse is used as a very close representative of humans. So by doing genetic manipulations in mice, especially with cancer type genes involved in cancer, you develop them in mice by inducing the mutations and then studying how the cancer can be treated in mice or how it develops. And then from that knowledge, you will be able to maybe cure human cancers, right? So the mouse is a representative model organism, easy to breed relatively and cheap. And ethically, not many people complain when you sacrifice mice. But obviously, sometimes you can't have a substitute. You have to work on our species. So volunteers are used under strict conditions, and they're very hard to maintain, very hard to control, expensive, and it takes a lot of money, right, in terms of setting up the trials and monitoring them and doing it correctly according to regulations. So even though other model organisms exist, sometimes the research has no alternative but to ask for volunteers in humans. So we are a model organism too for understanding how we primates function, right? So hopefully this long introduction to model organisms is enough to make you understand that size, expense, maintenance, and then relationship, biochemical relationship between different trees, between different branches of the tree of life, allow us to understand how the whole tree works. So that's why I took some more time than I normally do. So the final slide is this one called the C-value paradox. So as I said in the introduction, humans like to think we're the best of the best on the planet, right? Unfortunately, we're not. There are other creatures that have more DNA than us. There are other creatures that have more chromosomes than us. There are other creatures that have bigger cells than us. There are other creatures that can do more than us. There's other creatures that can see more colors than we can see, right? So we are sort of okay. We're okay. Although we are controlling the planet, but there's other reasons for that. Just opportunity and chance and luck, right? But what this table here is showing you is on the y-axis is telling you the size of the genome. What is the genome? That's the total DNA content of the cells of that species. So if I took a skin cell and I looked inside it, I'd find DNA in the nucleus, and then I'll find some DNA in the mitochondria, right? If I took all that DNA and I looked at it in the computer, I would have a sequence. And that would be my genome. And you would have your genome, and other creatures would have their own genomes, right? So the genome size is measured for one of each chromosome. So we call that the haploid genome. So when you have two sets of chromosomes, one from your mother and one from your father, you can't count both of them. You only count one. So your father's chromosome is taken and then is used to calculate the size of your genome, right? So that's what we have on the y-axis. Now on the y-axis, the other thing is the scale is going up logarithmically. It's not a linear scale, right? So the distance between here and here is 1,000. The distance between here and here is 100,000. And the difference between here and here is 1 million, and then 10 million, right? So this is a logarithmic scale. So remember that. And then on the x-axis at the bottom, we just have bars corresponding to different types of creatures on the planet, right? So we have bacteria here. We have the non-living viruses here. And we have the fungi there. And then we have the protists right here in blue. And then we have the plants right here in green. So that's a green sort of plants, okay. And then we have insects, which are animals, over here. And then we have mollusks, sea creatures, shells, shelled creatures, mussels and things like that, oysters. And then we have fish over here. We have cartilaginous fish. These are fish that have cartilage as their skeleton and not bone, right? So that'll be things like sharks. And then we have bony fish, like a carp. And then we have amphibians, like frogs, salamanders. Then we have reptiles, alligators and things like that, right? Then we have birds. And then over here we have mammals. So whoever drew this figure was biased, right? They put mammals at the end, hopefully being the best. But let's look at the data. The data suggests that viruses, on general, have small amounts of DNA across all the viruses that we've studied, right? So some viruses have over 100,000 base pairs of DNA. Other viruses have only a few thousand, right? So viruses naturally are tiny little things, and they can't package too much inside their shells. Bacteria. Bacteria are small cells, they're prokaryotes. So naturally, the amount of space available inside their cytoplasm is not enough to carry too much DNA, although some of the larger bacteria can carry 10 million base pairs of DNA, right? But the average is of a couple of million. So E. coli will be about here. I think it's 5 million or something for E. coli. Fungi, they range, as you can see from the gold bar, from one gigabyte. Gigabyte. No, not gigabyte, gigabases, right? So what is a gigabyte? It's so megabyte is a thousand, a million, what is it? So you need to know this. So in your book, at the beginning, there's a SI prefix that relates everything together, right? So make sure you understand that. So a gigabyte will be a billion, right? So let's look at humans. Well, the humans are right here. They're on the lower side of the mammals. So look at all the mammals and look at their DNA content. Humans are not top of the range. They're near the middle, towards the bottom actually. And this red line here that I drew indicates that humans are about 3.2 billion base pairs, right, of DNA inside a human species. So you have 3.2, I have 3.2, Mr. Joe over there has 3.2, right? But you can see there are many other creatures on the planet that have more than us. Look at this protist, an amoeba, a single cell that you can't even see with your eye. It has 100 times more DNA than you do, 100 times. That is the paradox. Why? We're not sure. It could be that different branches of the tree of life had different accidents. And in the case of the amoeba, it just flourished. In the case of our branch, it stayed steady. I don't know. That's why it's a paradox. So we see what we see, and we're trying to figure out what happened in the past, right? And plants, some plants have much more DNA than we do, all the mammals. Insects, some insects have more DNA than you and I, right? You can see most fish of this cartilaginous type have more DNA than humans do. And amphibians are way up there too, right? Certain types of frogs and things like that. So you can see we're beaten by a lot of categories in the content of DNA that our cells have. So that's what we call the C-value paradox, all right? So half an hour for this lecture, maybe a little bit more, that's appropriate. So I thank you very much. And then the next video will be on chemistry. Thank you, bye-bye. See you next time.